Students, this class is for on the request of certain students, those who did not follow the process of digestion properly. So I am giving the same process of digestion and digestive glands once more in this topic. And listen carefully and make sure you have understood it. Okay? Digestive system consists of digestive organs and also the digestive glands. Digestive glands produce their secretions which in one or other way helps in the digestion of food. Apart from large number of gastric and intestinal glands present in the lining of the stomach and intestine, there are three main associated digestive glands which pour their secretions into the alimentary canal. They are the salivary glands, liver and pancreas. So you can see together all these are the digestive glands. Of these, the three main important digestive glands are salivary glands, liver and pancreas. The other two are gastric glands and intestinal glands. First we will see about salivary glands. Salivary glands, there are three pairs of salivary glands. They are the parotids situated in the cheek, submaxillary or submandibular in the lower jaw, sublinguals below the tongue. There are these three pairs together secrete the secretion called saliva into the buccal cavity. So these three are the three pairs of salivary glands. And this secretion of salivary gland called saliva is watery and contains the enzyme salivary amylase along with mucus and other substances. So this saliva contains electrolytes like sodium, potassium, chloride, bicarbonate and enzymes like salivary amylase and lysozyme. Two enzymes are salivary amylase and lysozyme. So saliva not only helps in digestion of food, but it moistens and lubricates the food to make it easier to swallow. So it helps to mix the food with saliva with the food and helps in the swallowing. So these electrolytes and enzymes are present. The enzymes you can see salivary amylase enzyme. Salivary amylase enzyme converts the complex starch, that is starch is a complex carbohydrate, into simpler maltose, simpler substance called maltose, or we can call starch is a polysaccharide, which is a polymer which consists of many sugar molecules, and it will be broken down into simple sugars which consist of lesser number of sugar molecules. At last finally it will be converted to the simpler form. So in the salivary amylase enzyme converts almost 30% of the carbohydrate into disaccharide, that is example is maltose. Then lysozyme, it kills the bacteria, or it is having an antibacterial property. Then next, so this is the composition of saliva. You can see the composition of saliva. Next gland is the liver. Liver is the largest gland in our body. It weighs around 1.2 to 1.5 kg. And it is situated in the upper right region of the abdomen, just below the duodenum. Next digestive gland is the liver. Liver is the largest gland in our body. It weighs around 1.2 to 1.5 kg. It consists of two lobes and it is situated in the upper right region of the abdomen, just below the diaphragm. And the liver contains two lobes, and these lobes again contain lobules called hepatic lobules, which are the structural and functional unit of liver because they contain the liver cells or hepatic cells. So you know, cell is the structural and functional unit of an organism. So all organisms are made up of cell, and here this liver consists of liver cells, and it is present in the hepatic lobules, and they are also called hepatic cells. Each lobule is covered by a thin connective tissue sheath called Glissen's capsule. So each hepatic lobule is covered by thin connective tissue sheath called Glissen's capsule. So this is a short account on the structure of liver. Now coming to pancreas. Pancreas is a compound gland, and it is situated between the limbs of duodenum, and it is both endocrine and exocrine gland. So the exocrine portion secretes alkaline pancreatic juice, which contains enzymes that help in digestion, and endocrine part secretes hormones, insulin and glucagon, which helps in maintaining or balancing the sugar level in the body. So pancreas, another important gland. The endocrine part doesn't have function in digestion. It is the exocrine part, that is the cells which secrete pancreatic juice. This juice is having a role in digestion since it contains enzymes. So pancreatic juice is secreted by pancreas, exocrine portion of pancreas, which contains pancreatic enzymes. Now we'll come to the duct system of liver, gallbladder and pancreas. So this is the duct system, where you can see this is the pancreas, and liver is not shown here. From the liver the ducts are coming, and this liver produces the secretion called bile. So liver produces bile, and this bile is temporarily stored in gallbladder. Then you can see this is the U-shaped duodenum, first part of small intestine. So this is the duct system which actually carries the pancreatic juice and the bile into the duodenum. So we'll see the duct system. So here you can see hepatic ducts. These are the ducts arising from liver. Hepatic ducts carry the bile, and this bile will be temporarily, it will be stored in the gallbladder. And this duct, this one coming from the gallbladder and joining with this is called the cystic duct. It is called the cystic duct. So through the cystic duct, this bile will be stored in this, and the cystic duct and the hepatic duct together joins to form this duct called common bile duct. So the common bile duct passes towards the duodenum, where on the way it is joined by the pancreatic duct coming from the pancreas, and this common bile duct and the pancreatic duct together, it will be joining to form this green color portion. This is called hepatopancreatic Hepato is from the liver and also from the pancreas. So both the secretions are coming through this hepato-pancreatic duct, and they will join into this, open into duodenum. Okay, so this is the duct system. And so you can see gallbladder is here where temporarily bile is stored here. So hepatic ducts carry the bile from the liver and it will be stored in the gallbladder through the cystic duct. Then when this bile is needed for digestion, it will pass through the common bile duct and it will join with the pancreatic duct to form hepato-pancreatic duct, and then it will open into duodenum. And the bile duct and the pancreatic duct open into the duodenum as the common hepato-pancreatic duct, which is guarded by, here there is a sphincter called a sphincter of Oddi, which regulates the passage of substances through this. Now coming to digestion of food. Digestion of food. So we'll start with the digestion of food in step-by-step manner. Digestion is both a mechanical and a chemical process. It starts from buccal cavity, and about 30% of starch is hydrolyzed to maltose or disaccharide by salivary amylase. So starch is actually nothing but it is polysaccharide, and it will be broken down into a disaccharide containing, di means 2 sugars. So poly means more than too many sugar molecules together form a polymer or polysaccharide, that is starch. Starch will be broken down into disaccharide. It consists of 2 sugar molecules. So this much digestion, almost 30% of the starch is digested to maltose or disaccharide by salivary amylase. That is the reason why we should chew properly the food when we eat, so that 30% digestion should take place inside the mouth. Otherwise there will be overburden for the rest of the digestive system. Then coming to buccal cavity. So inside the buccal, so from the now we started with the process of digestion. Inside the buccal cavity, mastication, 2 things are happening: mastication of food and facilitation of swallowing. So inside the buccal cavity, mastication of food and facilitation of swallowing. So mastication of food is the teeth. Actually, there are 4 types of teeth. These teeth will chew the food and mix it with saliva. So the teeth and the tongue with saliva masticate and mix up the food. And mucus in saliva helps in lubricating and adhering masticated food particles into a bolus. So inside the mouth, the teeth will chew the food, and that partially chewed food will be mixed with saliva by the tongue, and the mucus in the saliva helps in making into a structure called bolus. So this partially digested food particles will come closer to each other and form adhere. Adhere means to come closer to each other and form adhering masticated food particles into a bolus, into a structure called bolus. This bolus passes to pharynx, next part of the digestive system, and then to esophagus by swallowing, or there is a term for swallowing called deglutition. So by the process of deglutition or swallowing, this bolus is pushed into the pharynx. So this is done with the help of tongue. So in this way, food first changes to bolus, then it is pushed into the esophagus, pharynx and the esophagus. Then the lysozyme in the food saliva prevents infections, and salivary amylase, you can see, converts starch to maltose. Maltose is a disaccharide. Then stomach. Next, stomach. So now we discussed about the digestion in buccal cavity. Next is stomach. Stomach, the mucosa of stomach. So you know what is mucosa? 4 layers of the alimentary canal, wall of the alimentary canal: outer serosa, muscularis, submucosa, innermost is called mucosa. So inside the stomach, the mucosa of stomach has gastric glands which contain 3 types of cells. So mucosa of stomach has gastric glands that contain 3 types of cells called mucus neck cells which secrete mucus, peptic or chief cells which secrete pepsinogen, parietal or oxyntic cells which secrete HCl and an intrinsic factor. We'll see what are these things. So the mucosa of stomach has gastric glands which contain 3 types of cells. Mucus neck cells or secrete mucus. Peptic or chief cells secrete pepsinogen. So actually this pepsinogen is an inactive enzyme. Inactive protein digesting enzyme capable of digesting protein. And this pepsinogen gets converted to an active pepsin. Pepsinogen is the inactive form converted to active pepsin in the presence of HCl. So you can see the third one, cell HCl secreted by parietal or oxyntic cell. So in the presence of HCl, pepsinogen gets converted to pepsin, which is an active protein digesting enzyme. Then the mucus neck cells secrete mucus. This mucus protects the inner lining of stomach from the harmful action of HCl. You know, HCl is a strong acid. So due to this, the intestinal wall or stomach wall can be damaged. So this mucus helps or protects the inner lining of the stomach. And an intrinsic factor also is produced by parietal or oxyntic cells which helps in the absorption of vitamin B12. So now you can see inside the stomach what are the things happening. Bolus reaches the stomach and mixes with the acidic gastric juice to form chyme. So bolus mixes with acidic gastric juice to form chyme. That is a partially digested food inside the stomach. It is called chyme. Now this pepsinogen is a proenzyme. It is an inactive enzyme. This pepsinogen is converted in the presence of HCl into pepsin, which is an active enzyme. Then what does this pepsin do? In the presence of pepsin, proteins are converted into proteoses and peptones. These two are the simpler form of proteins. Not the simplest, but simpler form. Intermediate forms of proteins. The simplest form of protein is amino acid. So before that, proteins are broken down into proteoses and peptones. So proteins are large chains of amino acids. Many, just like beads on a string. So many beads, if you are putting on a string, so that beads you can imagine this like amino acids. And that complete one is a protein, and each connection we can call peptide bond. That you will be learning again in another chapter. So proteins, they are long polypeptide chains. That is, they are called polypeptide chains because they are made up of more than many peptide bonds. So proteins, they are broken down in the presence of pepsin into proteoses and peptones. Next is small intestine. So the partially digested food from the stomach now reaches the small intestine. Small intestine receives 3 secretions, one from the liver, one from pancreas, and another from the intestine itself. So liver secretes bile, then pancreas, pancreatic juice, intestine, intestinal juice. So all these 3 secretions are secreted into the small intestine. So first the pancreatic juice. So pancreatic juice consists of enzymes called trypsinogen, chymotrypsinogen, procarboxypeptidase, amylase, lipase, nucleases. So these enzymes are present in pancreatic juice. Trypsinogen, chymotrypsinogen and procarboxypeptidase, these 3 are protein digesting enzymes, and these three are in an inactive form. So these three enzymes are protein digesting and these three are in an inactive form. So in order to change into an active form, it happens in the presence of another enzyme secreted from the wall of the intestine. So understand these three are protein digesting enzymes. Amylase, of course, it is a carbohydrate digesting enzyme. Lipase, lipid digesting or fat digesting enzymes. Nucleases, which digest the nucleic acids like DNA and RNA. So these are the pancreatic juice enzymes containing in the pancreatic juice. Now we'll see how the three enzymes are getting activated. So these three enzymes in the presence of another enzyme called enterokinase. Enterokinase is actually secreted from the wall of intestinal mucosa. So innermost wall of the intestine called the mucosa layer actually secretes an enzyme called enterokinase. This enterokinase activates trypsinogen to trypsin. So you can see trypsinogen, actually this is converting trypsinogen to trypsin, which in turn activates the other two enzymes. So these are the inactive enzymes: trypsinogen, chymotrypsinogen, procarboxypeptidase. These three are inactive enzymes. These three get activated into trypsin, chymotrypsin, carboxypeptidase. So these, in the presence of enterokinase, this activation happens. So enterokinase secreted from the wall of intestinal mucosa activates inactive trypsinogen into active trypsin. This trypsin in turn activates other proteolytic enzymes. So these three are called proteolytic, means protein digesting enzymes. Now we can see these are the steps which are happening in the small intestine with the help of the secretions of pancreas. So pancreatic juice helps in the digestion of all these. So first you can see pancreatic juice digests proteins, peptones, proteoses into dipeptides. So in the presence of this activated enzymes, trypsin, chymotrypsin, carboxypeptidase, the proteins, peptones and proteoses. So all the proteins may not be converted into peptones and proteoses inside the stomach. So the rest of it will be converted, then peptones, then proteoses. So all these three will be again converted into next similar form called dipeptides with one or two amino acids. It is called dipeptide. So peptide means that is the name of the bond which is found between the amino acid, peptide bond. Then polysaccharides or starch is converted into disaccharides in the presence of amylase. So polysaccharide, starch is a polysaccharide which is converted to disaccharide in the presence of amylase enzyme. Then fats gets converted to its next simpler form called diglycerides in the presence of enzyme lipase. So fats get converted to diglycerides, which again gets converted to monoglycerides. So in this way fat digestion also takes place. Then nucleic acids. Nucleic acids in the presence of enzyme nucleases will be converted to nucleotides, which in turn will be converted to nucleosides. So nucleotides, they are the next simpler form in the nucleic acids, which are further broken down, which can be further broken down into nucleosides. This nucleosides again needs further digestion to its simplest form, that is done with the help of intestinal juice. So here, with the help of pancreatic juice, nucleic acids will be broken down into nucleotides, and in turn it will be broken down into nucleosides. Nucleic acids are the DNA and the RNA, deoxyribonucleic acid and ribonucleic acid. So these are the genetic material, and this will be broken down into its simplest forms. Next, monoglyceride, nucleic acids, nucleosides, same thing. Then coming to bile. Bile consists of the following. Bile consists of bile pigments called bilirubin and biliverdin. And bilirubin and biliverdin are bile pigments. It also consists of bile salts, cholesterol and phospholipid. So these are the contents of the bile. And bile helps in emulsification of fat, and also it activates lipases. So bile is the secretion of liver which is concentrated and stored in gallbladder, and until it is needed it will be stored in the gallbladder, and it is poured into the duodenum, and it consists of the following. Then bile does not contain any enzyme, but it is extremely important in the process of digestion, and as it helps in the emulsification of fat, that is breaking large fat or oil droplets into small droplets. So why this is broken down? When it is broken down, it increases the surface area available for action of lipases. So it activates, actually lipase is the enzyme which breaks down fat. Bile helps in breaking down of the fat into small droplets so that lipases can easily act on this fat, small fat droplets. Okay, so this is about the structure of digestive glands and the process of digestion up to pancreatic juice. So you just revise this once more, just read it properly. Notes, also go through the notes, and again you understand step by step, you study these points what is given here. Then in the next class I will be giving the secretion, that is intestinal juice, and the enzymes present in it, and the process, how these nutrients are, how these forms are hydrolyzed into the simplest forms with the help of intestinal juice. Okay. I hope with this your doubts are clear. You have clearly understood the process of digestion. So go again, pause it and see it, and make sure you have understood this, and try to answer the questions and study the notes properly. Thank you.